Hi, my name is Susan Yee. I'm from University of Delaware, that's located 2 hours north of Washington, D.C., and 1 hour south of Philadelphia. And thank you for the opportunity to be here to talk about my research that I've been doing at Delaware. For the sake of brevity, I'm going to kind of skip over this slide only because I think we all know what biochar is and what it and its benefits. My research has been mainly focused on looking at soil water retention in biochar. The major problem with biochar is that its effect on soil water retention is very unclear. Therefore, predicting the biochar's effect on soil water retention at larger scale is very difficult, particularly for computational modelers who wants to understand its greenhouse gas effects and also irrigation volume calculations. Therefore, our study, we looked at all the factors that are affecting water retention relationships. So we listed all the different factors, and we tried to eliminate some of the factors that are affecting water retention, such as texture, that is related to the interparticle pores, which is voids between the particles that affects capillary water. Microporosity, which is affects the intraparticle pores, which are the particles within the pore volume, also affecting the capillary water. The surface area, which affects absorbed water in the dry end of the water retention curve. Wettability, which has to do with certain carbon interactions and leachable organics and some aggregates. And all these factors can change over time. But if we know the biochar's effect on each of these factors, we can kind of predict biochar's effect on water retention. So in our lab, what we did a very controlled fashion to test the effects of biochar on microporosity particularly and on surface area and how that increases water retention. So the materials that we used was homogeneous type of soil, sand that has been sieved to between the sieve sizes 0.5 and 0.9595, and sandy loam, and that was mostly picked because it's a prevalent soil in Delaware. We used 2 different biochar, poultry litter biochar, also sieved to the same particle diameter. Soil reef, which is pine wood biochar, which is commercially available, that was produced at 550 degrees. And it was also sieved. But what we also did was we rinsed our biochars to remove all the osmotic effects and any kind of organic coatings, and we also removed salts on the surfaces. So what we did was we basically had a control, both sand and sandy loam. We looked at the particle size distribution. We tried to remove any kind of textural effects that we were going to see. Sorry. So with our control, we had 2 controls. Sorry, 2 controls, which was sand and sandy loam, 2% biochar, which was either poultry litter biochar or soil reef biochar, and 7% at 2 of those mixtures. And you could see when we did a particle size distribution, we saw a minimal effect on texture. So we continued with our experiment. And we also tried to look at the geometric effects of SEM images, and it clearly shows that soil reef biochar was more porous than the poultry litter biochar when it was sieved to the same particle size here. So because of this, we wanted to continue our study with characterizing our surface area and intraparticle volume and how that will affect our water retention curve. So typically we did 2 different measurements, nitrogen gas, BT surface area, mainly because of the standard methods in most of the soil papers that we've read, but then it only characterizes the pore diameters within this mesopore region. You could also use, I think, SEM, and we have SEM images. However, you need to use ImageJ programming from NIH, and it's very subjective because it actually uses different color schemes, and we didn't pursue that any further. So we ended up exploring mercury porosymmetry, which is basically a non-wetting fluid, and we incrementally injected mercury at different pressures to calculate the intraparticle pore volume of both of our biochar samples, our soil reef and our poultry litter biochar. And we ended up also collecting the water retention curve, a drainage curve using these 3 different instruments, the tension table that measures the saturated water content, and all the way up to the air entry point, the pressure plate extractor to get the capillary region of the water retention. And also we used this WP4C dew-point potentiometer that measures the absorbed end of the water retention curve using the Kelvin equation. And we use this capillary pressure, and we plotted our log capillary pressure over the volumetric water content, only because it actually shows a very clear hysteresis of the water retention curve. And our results show that for the nitrogen gas BET surface area, there's a huge difference between the poultry litter and soil reef biochar. And then the intrapore volume were similar to, I think it was about a difference of 20%. But once we get the mercury porosimetry data, we had to process it even further to differentiate the difference between the inter and intraparticle pore volumes. And there's a method that you could read that actually helps you differentiate these two by looking at the inflection points of these of the data. And here's a very clear particle pore diameters. And we were also interested in trying to obtain this pore diameters, mainly because the capillary water content is the capillary equation is related to the pore radius. So once we obtain this, maybe we could potentially calculate the capillary pressure. And this is the pore volume that we have also obtained from the mercury porosimetry data. So here are the results. So this is the poultry litter biochar mixed in sand. This is the poultry soil reef biochar also mixed in sand. This dotted line is the mercury porosimetry data that is plotted on this secondary axis along with this pressure, and same thing is done here. And we only plotted the 7% poultry litter biochar mixed in sand and also in soil reef 7%, just to see a consistency in certain patterns and how biochar is affecting this water retention curve. And this is a water retention curve of our reference soil, sand alone here. And then this is our experimental results. And we see that this end between the PF pressure of 4 to 7 that's collected using the dew-point potentiometer, the water increase is due to absorbed water on the biochar surfaces of the poultry litter. And we have assumed that only because the mercury porosimetry data at different pressures are nearly 0 at this region. But then this is the intrapore particle where the water is being held within the pore sizes, because it clearly shows that it drains, and it also shows in this region as well. And we are assuming that there is no effect in water retention here, only because the difference between the reference soil here to the 7% poultry litter biochar mixture is a net effect between the intraparticle pore volumes plus the absorbed volume. And we're seeing we were able to calculate the same consistent effect in this region. And this is due to absorbed water. This is due to intraparticle pore draining until it becomes 0. There is no net effect here because this is basically intrapore particle plus absorbed water. And the values here, the difference from here to here, here to here, is basically a total intraparticle pore volume. And it's kind of amazing we were able to actually see that by looking at different mixtures of sand and biochar. But once we looked at the sandy loam, it was a little bit unclear because this is a natural soil that we collected in southern Delaware. It shows that there's a very small effect. You will still see an increase in absorbed water here. It's just, you just can't see it because it's transposed on top of each other, and the experimental points are a little bit bigger. We're seeing the same effect here, but then here what we're seeing is that there's a lower increase in water retention compared to the soil reef, and we didn't quite understand what was happening. So what we did was we developed a mathematical model to understand what's going on. So based on simple mass balance and ignoring any kind of textural terms, we developed this model to calculate the volumetric water content, and that is the relationship between the volume of water in the reference soil at various hydraulic pressures, capillary pressure, plus the volume of water that would be held in biochar at different pressures times the fraction of the biochar. For our case, it was 2% or 7%. And this volume of water that's held in the biochar is the intraparticle water at different capillary pressure plus the absorbed water at the higher pressure end. And this total volume was the volume of our core sample. And our model predictions actually work pretty well for our sand. So here's the 2% data. Here's our reference soil. And at 2%, we're able to basically predict the water retention curve at 2%, 7% for poultry litter biochar mixed in sand, and also with soil reef biochar here at 2% and 7%. There was a slight underprediction in the really adsorbed end. I don't quite understand what's happening here. It could be due to some kind of desorption, adsorption, isothermic reactions. I don't quite understand it. And that's actually our for future research. But once we looked at our sandy loam, it actually predicted better than our sand mixtures. So this is our reference soil, this 2% and 7%. It kind of over-predicted for our poultry litter biochar mixtures, but with our soil reef biochar mixtures, it did a pretty good job on predicting this water retention curve. So there are certain limitations to the model that we have developed. We didn't consider any kind of aging effects, so we're not quite sure whether if we use this model on natural soil, especially in high clay content soils, I don't think the model would work because there could be some particle clogging, there could be some kind of mineralization and hydrogen bonding that could occur. There's some aggregation, and also there's some particle fragmentation because biochars are very sensitive to just any kind of breakage or even like through weathering. We didn't even consider any kind of textural effects, and then we didn't also consider soil water repellency. But we believe that the model could be developed even further with time. So the conclusion of our study was that absorbed water in biochar, it increased water at permanent willing point up to 5 times for sand, but only 20% for sandy loam. Intraparticle water in biochar increased water at field capacity up to 50% for sand and 25% for sandy loam. And the model predicted water retention reasonably well for both biochars, both biochar mass fractions, and both of those soils, except for the absorbed end of the water retention curve. So I'd like to acknowledge Robert Hill, who let us borrow the instruments that we collected for our study, and our funding agencies. Thank you.
Thank you very much, Susan. Maybe a question before we wrap up? Anyone? Yes, sir.
I think it has to do with the size of the particle pore volume of different biochars. So what we did was we characterized a pore volume size of 2 different feedstock sources, poultry litter and pine wood biochar. And we saw that the pore volume was higher for soil reef biochar, and it was able to actually hold more water. I think it has to do with certain pressure effects, but our research was based on a drainage curve. So if there's a wettability curve, I don't know whether the model will even work, or whether at a certain pressure the water will infiltrate or penetrate through certain very small pore volumes. I don't know if that answers your question.